Okay, welcome everybody to our Hutch at Home series. Don't forget about your HPV vaccine. We're really excited to have Dr. Denise Galloway here with us today. I'm Jeannie Chowning, Senior Director of Science Education at Fred Hutch, and welcome everybody. I'd like to acknowledge that we're on the traditional land of the first people of Seattle, the Duwamish, past and present, and honor with gratitude the land itself and the Duwamish tribe. And feel free to introduce yourself in the chat. Please stay muted during the presentation. We'll address questions during the Q&A, and please sign up for our newsletter to receive news of upcoming events. And there's a QR code down there in the bottom right. And hope that you can join us at future Hutch at Home events. Okay, so really glad to have Dr. Denise Galloway with us today. Dr. Galloway is a scientific director of the Pathogen-Associated Malignancies Integrated Research Center at Fred Hutch, also a professor of human biology in the Human Biology Division and professor in the Public Health Sciences Division at Fred Hutch, and working with 2 viruses that cause cancer. And I'm really excited to hear more about that. A lot of people don't appreciate the role of bacteria and viruses in causing cancer. So this is something that I think is really interesting to hear more about. So I am going to turn it over to Dr. Galloway. So go ahead and feel free to share your screen, Dr. Galloway.
Okay, oops. No.
There you go.
There we go. Okay. Well, I thought that there's been so much talk about the SARS-CoV-2 vaccine that I didn't want everyone to forget about other vaccines that are necessary. And this one is particularly important for adolescents. So I will tell you about it. And Jeannie asked me to first say something about myself and how I got to where I am. So I will do that. This is for those of you who don't know New York City, this is the subway map of the city. And the part up at the top, let me get my pointer. So this borough up here is the Bronx, and I was born and grew up here. And oops, wrong way. And then I went to high school here. One of the key elements was that I could take the subway from my house down to the school, the high school. And it was this great Gothic looking building on Lexington, which was really a great place. It was all girls at the time that I went. And all smart girls. You had to get tested to get in. And I think that did a lot for my confidence, because many girls, when they start high school, sort of lose their desire to speak out and to sound smart. So then I went to college further up. And after that, I went to graduate school back down in Manhattan. So I was born in the wool New Yorker. And then once I finished my PhD, I did a postdoc out here. This is Long Island. And I did a postdoc at Cold Spring Harbor Laboratory, which is sort of the opposite of the Gothic building on Midtown Manhattan, but it's very rural. It's about 35 miles outside of the city. And it's really an exciting place for molecular biology. They have summer meetings and courses and just great people who were there to work with. So when I was there, I started my love of what became my life's career. So what I was intrigued with when I was a postdoc was the fact that you could take a virus, and in fact, you could just pull out some genes from the virus and put them into a normal cell, and it would transform them into something that was more like a cancer cell. And that really excited me, and I stayed with it. Now, when I was a postdoc, we were doing this on a model system. SV40 is a monkey virus, and it has this property of being able to transform cells. But I was interested when I got to my own lab in trying to work on a system that was more medically relevant. And I started working on herpes viruses because at the time people thought that's what was involved in cervical cancer. But it turned out not to be. And in the early 80s, a group in Germany discovered that human papillomaviruses were in cervical cancers. And so I switched and started working on HPV. So to start my own lab, we drove across the country to Seattle, to the Fred Hutch Cancer Center. This is what it looks like now, not what it looked like when I moved there. But we really have great facilities, great lab buildings, and other things here at Fred Hutch. So let me tell you, I'm going to tell you today about what I do, and particularly how the vaccine was developed and what it does. So this is the viral genome in a simplified form. It encodes just 8 genes. And these 2 genes, E6 and E7, turn out to be what's responsible for making a cancer cell. And in all of the cancers that are associated with HPV, I'll tell you more about that in a minute, these E6 and E7 genes are integrated into the host chromosome and are always expressed, and they are what drives the tumorigenicity of these cells. Now, the vaccine story that I'll talk about today involves one of the 2 capsid proteins. L1 and L2 are what make up the virus capsid. And you can make something that looks like the viral particle just with L1. And that's used to vaccinate and generate neutralizing antibodies. So when I talk about HPVs, it's actually a very large family of viruses that cause all kinds of things. Plantar warts on the soles of your feet, common warts on your hands or your body. The beta papillomaviruses are all over your skin. But the ones that I'm going to focus on are the ones that infect the genital tract, particularly the ones that cause cancer. So there are 2 over here, the line's a little off, over here, type 6 and 11, that cause the majority of genital warts. Genital warts are common. They're difficult, can be difficult to treat, but they never cause cancer. They're benign. On the other hand, there are 2 groups of these viruses that cause cervical cancer and all of the other cancers that are associated with HPVs. So this is the normal life cycle of the virus infection. So the virus gets in through tears or abrasions in the epithelium, and it goes down to this bottom layer of basal cells. I really think it goes down to the basement membrane, which acts sort of like flypaper to trap the viruses. And then when the cells start to heal, they roll over it and become infected. So it's only these basal cells that can become infected by HPV. Now they have a mechanism, as I showed on the earlier slide, the viral genome is a circle, a double-stranded DNA circle. And there's a mechanism to transfer it to the daughter cells. So when this cell divides and gives rise to daughter cells, HPV hitchhikes along. And it's kept at a fairly low copy number in these cells, around 50 copies per cell. And then as these cells continue to divide and differentiate, once they get to the most peripheral layer of the epithelium, then the virus is capable of replicating a lot and expressing those capsid genes and forming new infectious virus for the cycle to begin over again. So that's what normally happens when you get an HPV infection. Now, occasionally something goes wrong. So you have a break in the viral genome. We always have breaks in our cellular genome. Every day, DNA gets broken by various sources. And if it integrates in such a way that those E6 and E7 genes are intact and gets expressed, then it causes these cells to proliferate, eventually without differentiating, and eventually that becomes a cancer. So if you think about the life cycle in terms of people, the population, you get infected with those types that cause genital warts or cancers or benign lesions when you first become sexually active. And very soon after that, there's a detection, you can detect HPV in the genital tract. We've done studies at the University of Washington where we followed college freshmen. First, we started with women, and we found after 2 years of sexual activity, about a third of them had HPV detectable in the genital tract. And among college men, about 2/3 had detectable HPV. Now, one of the manifestations of that, as I showed you on the previous slide, is instead of getting differentiated cells as you move up the epithelium, you get these undifferentiated cells. And this is called either cervical intraepithelial neoplasia or squamous intraepithelial lesions. And the more of the epithelium that gets filled up with those cells, it's called high-grade SIL. And if it's just a third of the epithelium or so, it's called low-grade SIL. And then once the whole epithelium is filled up with these abnormal cells, it's called prevalent carcinoma in situ. So it's not cancer yet because it's still all within the epithelium. And you can see that the median age in the United States is about 30 years old. And then eventually it will progress if not treated, or at least most of it will, and become invasive cancer. And the median age for cervical cancer in the US is 49.5. So, fairly young people get cervical cancer. So this is just picturing it in another way. You have the normal cervix, it gets infected, and most of the time it gets cleared. About at least 90% of the time, your immune system is able to control it. Now, clearance, I think, is not quite the right term for it, because it just becomes undetectable by PCR. But everyone believes that there's latent HPV that remains with you. Now, when it's in this stage, where you can detect that you have an infection, the DNA remains as a circle, as I showed before, an episome, and you have a very low level of expression of these E6 and E7 genes. But now in again, a fairly small percentage of cases, it will progress and you'll have a precancerous lesion. But even these, about 30%, maybe about a third of them will regress. Again, normally your immune system can control it, but there's a great danger that these will go on to invade and form cancer. Now, in this state, the virus genome becomes integrated into the host cell DNA, and by various mechanisms, you turn on a much higher level of expression of E6 and E7, which, as I mentioned, are the genes that are necessary to drive the cancer. So of the many types of HPVs that I showed you before, about half of this orange block is HPV 16, and about half of all cancers are HPV 16 worldwide. The next biggest component is HPV 18, and together these two account for around 70%, slight geographic variations. And then there's a remaining about 15 types that, or 13 other types that can also cause cancer, but they cause a very small proportion of it. And again, it varies a little bit by geography. The other important thing to say is that nearly 100% of the cancers, and most people believe it really is 100%, and there's just some testing, errors or something. 100% of cancers, of cervical cancers, are caused by HPV. So throughout the world, the incidence of cervical cancer is the highest. It's higher than this graph shows. It's about 530,000 cases a year and about 275,000 deaths. Now, all these other sites, the anus, the vulva and vagina, the penis, and the oropharynx are also associated with HPV-associated cancers. It's just a slightly smaller percentage. Again, I think with newer testing methods, these percentages are a bit higher. And the interesting one, of course, is the oropharynx, which both men and women have. And it's really about half of the oropharyngeal cancers are HPV associated, and that number increases every year. Now, in the US, the picture is different. So cervical cancer is high, the highest among women, with vulvar, vaginal, penile, anal. Anal is about half male and half female. And I see, this has just moved over a little. Sorry, the graph has moved over. But this is oropharyngeal cancer. And so in the United States, HPV positive oropharyngeal cancer is higher than the amount of cervical cancer, because we have good screening methods to detect cervical cancer, and we don't have any screening methods to detect oropharyngeal cancer. And it's also 4 times more common in males than in women. Now, lest you think that cervical cancer is not a problem in the United States, here is a heat map of the darker the color means the more HPV associated cervical cancer is present. And you can see that in parts of South America, large parts of Africa, Asia have high rates of cervical cancer, where the US and Western Europe have much lower rates of cervical cancer. So, as I said, there are about 530,000 cases yearly and 275,000 deaths. However, the cost of screening and treatment in the United States is about $7 billion. So it's expensive to screen all the time and expensive to treat to reduce the burden of cancer. And so what we're hoping is that, or what we believe is that the vaccination and improving screening in other parts of the world will be able to reduce or completely eliminate the burden of cervical cancer. So how are we going to do that? now we're going to turn to how the virus was made and why it works. Okay, so when you think about vaccination, you could think about disrupting this paradigm in several places. One is you could prevent infection, and that's what most vaccines do, but not all. Secondly, you could mediate regression so that if you had a precancerous lesion, you could clear it by some type of a vaccination. Or if you developed cancer after you remove the cervix, you could use it to eliminate any residual disease that occurs outside of the cervix. Now, these two strategies are based on the idea that you can make a T cell response to those E6 and E7 proteins. Those are necessary for driving the cancer. They're expressed in the precancerous lesions at a high level. And clearly T cells are what normally mediates regression. And so you could try to make a therapeutic vaccine that could treat these different stages of HPV infection. There are many attempts for this that are ongoing, but there's nothing that's really that successful at this point. But on the other hand, preventing infection has turned out to be really a home run as a strategy. So how's that done? So this is what the virus actually looks like. This happens to be a cow virus, a bovine virus, but they all look exactly the same. So they have the L1 protein is all of this star-like projection that you see here, as well as the floor that connects all of them. So there are 5 of them that come together to form what we call a capsimer. And then there are 72 capsimers that come together to form this nice icosahedron. And for L2, there's only 1 molecule of L2 per capsimer. And we can't see it in this projection because in order for this to work, the way these reconstructions work is you take many images of capsids and you average them all to get a reconstruction. And when you do that, if things are not in a static position, if they're wiggling around, they won't show up on this type of EM. So the L2 protein sticks more inside. There's only a very tiny little piece of it that's on the surface. And although we used to think it was down the dimple of these capsimers, it's not. It's somewhere else with just a little piece sticking out. Now, inside of that is the viral genome. And the viral genome is a circle that's about 8 kb, and it's wrapped up tightly so that it fits in there in cellular histones, which are proteins that compact the DNA. So the breakthrough came when you could make virus-like particles. And so the way that you do that is you express the L1 protein. It can be produced in yeast or other mammalian cells. And 5 of these come together automatically to form the capsimer. And then if you incubate these capsimers or pentamers with a little bit of salt overnight and using the extract that's present in the cells that you express them, it folds up and forms something that looks exactly like the virus that I showed you on the previous slide. The differences are it only has the L1 protein in it, not L2. You don't need L2 to make this structure. And it doesn't have the viral DNA in it. So it can't cause cancer, can't express any of the viral genes because it's just this L1 protein that forms this nice icosahedron. So what I want to show you, this is now a crystallographic structure of one of those capsomers, just one of them. And if you take a slice through it, you see that all of these blue residues are conserved among all the papillomaviruses, which is why, as I said earlier, they all look exactly the same. Now, if you look instead face down, or look at the top of the capsomer, you can see in red, these are all variable. By that, I mean that the residues on HPV 16 are not the same as on HPV 18 or HPV 6 or 11. And so what your body sees is mainly what your immune system sees is mainly this surface. It doesn't see the conserved parts. And so the antibody response is very type specific. If you put in just an HPV 16 capsomer, then that's all of the response you'll get is to HPV 16, and those antibodies won't protect against the other HPV types. So the idea has been the following. Again, you start with a virus, you take out the L1 gene, and you can clone it into a vector that allows it to get expressed in cells and make more capsid, L1 capsid proteins. And then this assembles as an empty shell. And then you mix it with an adjuvant, which is something that makes it more immunogenic, and you vaccinate and you make antibodies. Now, as I'll tell you, the first vaccines that were made had 4 different HPV types in it. And the current vaccine that's in most use now has 9 HPV types in it. So around the same time, around 2006, these 2 vaccines, one made by GlaxoSmithKline and one made by Merck, became available. The bivalent vaccine has HPV 16 and 18, the 2 types that most commonly cause cancer. It's given in a 3-dose series. And more recently, we learned that actually 2 doses is just fine for adolescents. Adolescents make better antibody responses than people over once they've hit puberty. And so it's been shown that 2 doses of the vaccine are as good as 3. The quadrivalent vaccine made by Merck has 4 types in it, the 2 that cause genital warts and the 2 that are most commonly associated with cancer. And they're also given in this 3-dose schedule and a 2-dose schedule. And then I don't remember the year exactly, around 2012, I think, the 9-valent vaccine became available, called Gardasil 9, also made by Merck. And it contains 6 and 11, 16 and 18, and then the 5 next most common cancer-causing types. And again, the same sort of regimen. Now, one advantage that the bivalent vaccine has is it uses a very strong adjuvant, a very potent adjuvant, and it makes a good immune response, and it makes a little more cross-reactive antibodies than in the Merck vaccine that uses just alum. Okay, so back to this natural history study. So if you want to test if a vaccine works, you have to give it to people that have a chance of becoming infected. And so the efficacy trials that were done used initially young women and subsequently young men between the ages of 18 and 25 or 26. And you blindly do these vaccine trials, and you say, okay, I'm going to stop when there are 100 cases. And then you see, are the cases all among the people who got the vaccine or who got the placebo, or half and half? And that will tell you how well the vaccine is working. But that's not what you want to do, because you want to prevent infection. And these people that are in the efficacy trial are potentially can be infected because they're having sexual contact. You screen them initially to see that they are negative, but you want them to be having sexual activity so that some of them will become positive for HPVs. But the target population is in this 9 to 14 age group before they've initiated sexual activity. And the CDC actually recommends that you start it at age 11 and complete it by age 12 for the United States. So that's how the trials were done, but this was the target population. So the vaccine, so here's one of the earliest results just using the Gardasil, the quadrivalent vaccine. And you look for lesions, as I said, and these, remember, is cervical intraepithelial neoplasia, and you look for different grades or different severities. This is adenocarcinoma in situ, or you can look for genital warts. And so among those who got the vaccine, there were 4 cases of either HPV 6, 11, 16, or 18 lesions, in this case, 16 or 18, and 83 among those who received the placebo. And in the looking for genital warts, there was only 1 person who received the vaccine who got genital warts, and 91 in the placebo group. So it's really highly efficacious, 95 to 99%, which is great. Everybody's getting used to these kinds of numbers when you hear about the SARS vaccines, but if you think about the flu vaccine, it's about 50%. Now, how do you go from this study, which is in these 16 to 26-year-old women, to saying that it's going to work in children in the 9 to 14-year group? Well, you do something that's called immunobridging. So you say the amount of antibody that was generated to protect these women in the trial has to be at least the antibody level that would protect children. And so you do what's immunobridging. You do a study where you vaccinate children. You're not looking for efficacy because they're not sexually active. You're looking for an antibody response that's at least as good, if not better, than the group that are vaccinated in the 16 to 26-year-olds. And as I said, you generate a better immune response when you're vaccinated as an adolescent than as an adult. And so that's been the basis for the approval for vaccinating children as well. Studies were subsequently done in men, again, college, sort of this group of age group of men, and it was found to have very similar protection rates. And so the vaccine is now recommended for both girls and boys who are between the ages of 9 and 14, because although we think of HPV and cervical cancer, as I showed you, there's oropharyngeal cancer that's high in men. There is anal cancer that's reasonably high in men. And there's penile cancer, which is rare, but does occur. So what's next? This is what we're working on in my lab now. So what you want to know is, do you generate memory from the vaccine? And you want to know that because if you're going to give a kid who's 10 years old a vaccine and ask for them to be protected when they're 20, 30, 40 years old, you have to know that the vaccine will generate the type of memory that will last for a long time. So what is that and how does that happen? So here is a naive B cell. It hasn't seen anything it recognizes yet. And then it randomly, some of them will happen to be able to see the virus. And they bring it to a germinal center, which is like in a lymph node. And there it meets up with T cells, and it causes that cell to proliferate. And the ones that bind most strongly, they proliferate and they make antibodies. And the ones that bind the antibody most strongly are selected for. They amplify, they proliferate more than the ones that bind less well. And then class switching is something that is important in generating different classes of antibodies, like IgG, IgA, IgM. And so they undergo that maturation too. And then they give off 3 types of cells. One is within about a week after, or even less, of giving a vaccine, there are these short-lived plasmablasts that are in the peripheral blood. And what they do is they make antibodies that they secrete. So they're in the bloodstream early on. Then you also generate memory B cells. These memory B cells don't secrete antibody. They have antibody on their surface, and they go and reside in the bone marrow. And they're there, they come out, transiently a little bit at a time, so that if you were to get infected, now that memory B cell would see it and be able to start this proliferation all over again and make lots more cells that make antibodies. And the cells that make a lot of antibodies are these long-lived plasma cells. I think I said the wrong thing. These reside in the periphery as well, in the bloodstream as well. So these 2 are in the blood. And these long-lived plasmablasts go to the bone marrow, and they secrete antibodies. So the antibodies that are in your blood that are there to protect you right away from an HPV infection are made from these types of cells. And then in addition, you have these that get cranked up and will recognize the virus and start making lots more cells. And you go through this again and again. Now, what we've been doing is assaying these cells and these cells to try to see whether or not there's memory that's generated. And these cells, we haven't been able to study because we're not going to do bone marrow aspirates on normal, healthy volunteers. So we don't know about these cells, but we can measure antibodies in the blood for years after the vaccination is given. And from those studies, we've designed a way to pull out HPV specific memory B cells by using those L1 like particles that are the vaccine. We can pull them out for HPV 16 or 18 or any of the other types that we want. And we can show what they look like in terms of, are they affinity matured? And the way you do that is they will be mutated from what the germline sequence is. And are they class switched? And the answer to both of these is yes. You get HPV specific memory cells that are class switched and affinity matured. And that tells you that you're very likely to have a lasting immune response. Now, the vaccine preceding, the trials were done sort of preceding 2006. And so we have over 10 years of data in people who were vaccinated. And we know that there's still protection and there's still circulating antibody. So it looks like this vaccine is one that will last for life, sort of like your tetanus vaccine, not your tetanus, your smallpox vaccine, which some vaccines will last all your life, and others you have to get a booster every year or every so many years. And so we think this vaccine is really going to provide lifelong protection. So with that, I will show, this is an old picture. Nobody in my lab has been this close together for 1.5 years. So it's out of date, but it's a representative of most of the people that are still here. So with that, I'll be happy to answer any questions.
That was great. Thank you so much. So interesting. There was a question that came up in the chat earlier about how common are these dangerous HPV strains? So you showed.
Yeah, very common. So certainly, well, we know for HPV 16, it's the most common, and it's in about 10% of people. And I would say that if you take all of the ones that have any oncogenic potential, it's at least 30%. But for most of us,
30% of the people have
really,
those. Just
yeah, for most of us, it doesn't, you get an infection, you never know it. You have a pap smear, it may say you have an abnormal pap. And then if you go back, a year or two later, it's gone. Mm-hmm. So that's what happens for most of us.
Mm-hmm. I was wondering, what is it about the cervical cells or oropharyngeal cells that makes it so frequent at those sites?
Yeah, it's what's called a transformation zone. So there's 2 types of epithelium. There's the columnar epithelium, which is just 1 layer thick. And then there's the stratified squamous epithelium that's multi-layered, like that picture I showed you. And infection usually occurs right at the junction between those 2. Now, the virus can only replicate in the squamous epithelium, but sometimes it can probably, that's an area that can go back, it can switch its morphology back and forth at different times of life. So, for example, when you first start developing your period, you get some changes in that transformation zone as to where it is. And then when you have menopause, you have changes that go in the opposite direction. So it's really just those specialized junctions that are. Now, clearly it can infect other places. So there's a transformation zone in the cervix and in the anal canal, but certainly vaginal and vulvar is just squamous epithelium. And so I think it's not that can't be infected, it's just much rarer.
Mm-hmm. Mm-hmm. There's a couple other questions in the chat. How do you talk to folks who use this as proof that you can catch cancer from someone?
Well, I don't usually tell them you can catch cancer. I can say you can catch an infection that will cause cancer.
Yeah, so just kind of correcting their misunderstanding.
It's not that the cancer is infectious. But it's that the virus that's the trigger for it is infectious, and it's easily transmitted.
There's a question, have there been any obstacles from the multiple different types of vaccines available? I feel like before COVID, we weren't used to talking about different brands or types of vaccines.
Well, I think the world now is using either Cervarix, the bivalent vaccine, or the 9-valent vaccine, and they're going to stop making, or they have stopped making, the quadrivalent vaccine. In the United States, almost uniformly, it's Gardasil 9 that's used. And I think, certainly in the US, most people who don't work on HPV don't even know about Cervarix, because, if you say, it's time for your HPV vaccine, it's going to be Gardasil 9 these days.
Interesting. Feel free to unmute yourself if there are questions. Please ask questions. Danielle, go ahead.
I was just so amazed that my daughters could both get that vaccine.
Yeah.
And I just want to thank you, because this is amazing. This is absolutely amazing. And I promote it very heavily among my students at school. Luckily in Seattle, most people seem to be sane and they get the vaccine. So I do have a question. I'm just really wondering if there's anything known about the mechanism of that, of durability, because I understand that that's probably pretty hard to study. I know you said that the memory B cells, the long are really important. Are T cells also important for that? And is there anything known about how that works?
Well, you have to have what are called helper T cells to make that memory response in the lymph node. But if you think of, when most people think of T cells, they think of the ones that are cytotoxic, the CD8s, that would be what you'd want to generate to kill the tumor cells. But that's not, that's sort of irrelevant for the prevention vaccine. It does make, let's see if I can go back. Well, I won't. It makes CD8 cytotoxic T cells to L1, but that's sort of irrelevant, because by the time you're making a lot of L1, you're up at the periphery of the epithelium where it gets shed anyway. So the T cells aren't doing anything to protect you. Now, you need helper T cells, so you do stimulate a T cell response to make antibody, to allow those B cells to proliferate.
Okay. So like, if you think more broadly, I'm sorry to ask this, but this is just so fascinating. If you think about something like measles or HPV versus flu vaccines, why does one last so long and one doesn't?
Yeah, that's not really well understood. I mean, the flu vaccine probably lasts longer, but it's just that the virus changes. And so you have to keep, when you get a flu vaccine every year, you're getting different viruses.
Yeah, I was actually, I'm busy teaching immunology, so I'm spending more time than I should reading stuff. But I saw this morning that I think this has just come out, that flu vaccine really only lasts about 3 months. Then that antibody response is way lower. It's
lower, for sure. But if you take people who are vaccinated every year, they have actually a poorer immune response, because they do have antibodies hanging around from the previous vaccine.
Okay. Thank you.
Yeah, sure. The other thing I should say is that although Seattle is good at people taking the vaccine, I've looked at the state of Washington, the counties that they, on the public health page, they have the counties, the percent of vaccine uptake. In some counties, it's really poor. And so, we're lucky that we have a well-educated population, or that people are pushing it, but it's not true even all over Washington.
You mentioned that also the vaccines have the protection against the genital warts. Now, do the genital warts ever transform cells?
No.
So
there is a rare, I mean, you never say never in biology.
Yeah, right.
Because there's always sometimes. So there are laryngeal warts, which are really, they don't transform, but they're common, not common. They're a rare thing in the population, but they're always 6 or 11. And extremely rarely you can get an HPV-6 associated cancer, not of the larynx where they get it, but it seems like it's spread further down the respiratory tract, and it's like lung almost. But that's a very, very rare event. So I'd say no for warts transforming.
But they're included in the vaccine just because
Well, because actually
It's-
they're common and they're.
It just
you
kind
know,
of prevent something that's
and-
inconvenient.
Yeah,
and
and,
not
nobody wants to
attractive.
tell your partner, I've got genital warts.
Right.
So they included it. If you're cynical, you could say they included it to make it attractive to men when they first came out with the vaccine.
Other questions? We have a few minutes before we wrap up.
I guess when you said 10% HPV 16 is about 10%.
In the general population.
Okay, and then the 30%, was that of the HPV strains or?
Yes, or mostly what I meant was all of the ones that have oncogenic potential.
Oh, okay, okay.
So
Because there are a lot of other ones that infect the genital tract and don't seem to do anything. Don't cause warts, don't cause cancer. They're just there if you sample it and you test for all these types.
Okay, last question. How does that E6 and E7 work? What is what
Yeah,
does it do?
I thought about whether I should put that in or not, but I didn't know how complicated to make it. Okay, so there are two tumor suppressors that normally control cell proliferation. One is the retinoblastoma protein. And what that does is it inhibits transcription factors that are necessary to drive the cell into proliferation, into the cell cycle. And so the E7 protein binds to RB, brings it to a ubiquitin ligase, ubiquinates it, and then it gets targeted for degradation. This is in the high-risk HPVs, the one that cause cancer. Then P53 is another tumor suppressor that is important in normal cells. If they get various kinds of stress, they upregulate P53, and that will also inhibit the cell cycle from cycling. And the cell will either go into arrest or it will die by apoptosis. So the E6 protein binds to P53, it binds to a different ubiquitin ligase, targets P53 for degradation, and gets rid of it. So I think of it as if, you know, RB is like the accelerator, the pedal, and P53 are the brakes, and you get rid of both of them, and you just continue to cycle and cycle. Now, E6 does other things as well, and one of the really important things it does is it targets telomerase. You know what telomerase is? It's the enzyme that puts on the ends of the chromosomes. And they get shorter and shorter and shorter. So if you take normal cells and put them in tissue culture, they'll divide for a certain number of times and then they'll stop. And they stop because their telomeres get too short and it's recognized as a danger signal, so it stops. So for a cell to proliferate indefinitely, to be a cancer, it has to have telomerase. And so there are two parts of telomerase. There's an RNA component, and there's the protein that's the enzyme component. And the enzyme component, which is called TERT, is not expressed in normal cells, which is why the telomeres shorten and shorten. But E6 is able to target a repressor of TERT transcription so that now you've turned on TERT, and now the cells can proliferate forever. So when we do this in culture, we put E6 and E7 into a cell, you now establish a cell line that will grow forever. And so, for example, HeLa cells, which are well known, have the HPV 18, E6 and E7 gene in them. And so, as you know, they proliferate forever.
That's such an interesting connection. I
Yeah.
love how you just dropped all that knowledge on us, because that was fantastic.
That was fantastic. But what does the virus do with E6 and E7?
Well, okay, so this virus, so
You
viruses.
can kick me out soon.
No, I'm happy. This virus, so, there are lots of different kinds of viruses. This virus is small. It doesn't have any replicative machinery of its own. The only thing it has is a helicase that unwinds the origin, and then it needs everything else from the cell. So you have to be in a cell that's replicating in order to get the virus going, right?
Mhm.
It's in the cell cycle to get it its own genome replicated. So E6 and E7 are doing that to a little extent so that the virus can replicate.
Wow.
So does anyone know what the difference between E6 and E7 from a tumorigenic HPV strain is versus a non-tumorigenic HPV strain?
Yeah, so both of them can, so the non-oncogenic strains can also bind RB and bind P53, but they don't target them for degradation.
Yeah.
So they're missing the ability to bind to that ubiquitin ligase. Two different ubiquitin ligases.
That's fascinating. Well, thank you so much for this talk. It's
You're welcome.
yeah, I think I'm going to be replaying especially this last part, and
The questions were more.
that's great.
interesting than the talk.
We've got a science group here, a number of scientists here who are also involved in education. So that's great. And thank you so much. I have just a couple closing slides, so I'll share those.
Okay.
I also have a picture of your lab from.
Oh, that's a more recent one, but it's
Yeah.
still, more than a year and a half old.
Yeah. And so for folks who are attending, feel free to drop out some evaluation and feedback into that link, and that can give us some information about this, and I'll put that in the chat. We also have recordings of prior Hutch at Homes, as you probably well know, but these are both on our SEP events page and also can be found at the SEP YouTube channel, which is linked on this SEP events page. So I want to give a shout out for our next virtual field trip, which is on the 25th. And I'll also put the link for registering to this into the chat. Dr. Parkhurst is our neighbor in the hallway, neighbors in the Weintraub area. And so her lab studies wound healing using the model organism Drosophila. And there'll be some really interesting visuals of how they do that. And so encourage folks to sign up for that. We are going to have one more talk and field trip in June, and then we're going to take a break for the summer because of our summer programming. So thanks again to all of you who joined us, and thanks again especially to Dr. Galloway. That was awesome, and hope you all have a great rest of your day.
Thank you, Dr. Galloway.
You're very welcome. Bye-bye. You can always, you can email me if you have more questions.
We may follow up because we do have the curriculum group that's working on P53 related materials. We talked about that already. I don't think they've reached out yet, but I will encourage them to.
Okay. All right. Bye, everybody.
Bye.
Thank you.
Thank you.